All right, good morning, everyone. Thank you for your patience. If you want to still go ahead and enter in where you're from in the chat bar, feel free to do that if you just joined us. But I wanted to welcome you to our second webinar in our series for the Lilly Center. At the Lilly Center, our mission is to make the lakes and streams of Kosciuszko County clean, healthy, safe, and beautiful. And these webinars are a tool that we're hoping to use to equip you as citizens to have the tools that you need to work on your own lake to make it clean and healthy and safe and beautiful. Today we have our research specialist, Adrian Funderburg, talking about blue-green algae across the state and county. Just so you know, we're going to be stopping once in the middle of the webinar and once at the end to take questions that you might have had come up. In the meantime, if you just want to enter those in the chat bar as well, we'll make sure to go back and cover those mid-session and then at the end. So with that, I will let Adrian take over.
Alrighty, hope everybody can hear me okay. Like Caitlin said, I'm Adrian Funderburg and I work as the Research Program Specialist here at the Lilly Center. And a lot of what we do here, especially on the research team, is focused on blue-green algae. Today's talk will be about blue-green algae research and monitoring across the state and county. And Kasia Krzyzewski, who is a senior environmental manager at Idem, so down in Indy, unfortunately was last minute not able to be with us today, but she definitely helped contribute content. So I'll still be presenting on the information that she would have been giving today. But unfortunately she wasn't able to be with us. So with that, let's jump right to it. There we go. Alrighty, so we have 4 main takeaways for this presentation today, things that we'd like to equip you with and things that we hope kind of stick in your head are some of the most useful things to know with this presentation. Those things are what blue-green algae actually is, its relevance across the state of Indiana and the county of Kosciuszko County, how to identify a blue-green algae bloom if you see one, and what to do about it when you do see one. So we'll be covering each one of these things in turn together. We have a couple partners that we'd love to thank. Idem, of course, the Indiana Department of Environmental Management, and specifically Kasia for helping contribute some of that content and also for partnering with us on our work sampling blue-green algae and toxins and such. The K21 Health Foundation, which we owe huge thanks to for funding this research in particular and for caring about the health of our community in these ways. WACF and the Winona Lake Preservation Association for partnering with us on this particular webinar and such. Like Caitlin said too, if you have a question or want a clarification of something during the presentation, please type your question right into that chat box and we'll answer it halfway through the presentation or during the Q&A section at the end. Type it right away when you think about it though, because we love Q&As and we would not want you to forget about it by the end. So just pop those in there and Caitlin will be watching and kind of keeping track of those questions while we go. So right off the bat, let's learn about what blue-green algae is. Blue-green algae goes by 2 names. It goes by the name blue-green algae, which you are likely familiar with just in signing up for this webinar. It also goes by the name cyanobacteria. Both of these names describe exactly what these organisms are. Blue-green and cyan both refer to their use of pigments or color compounds to absorb sunlight and create food with photosynthesis, just like plants and green algae do. So they photosynthesize, but they aren't quite plants or algae either. We call them blue-green algae, the organisms that we are working with in particular, because they live in aquatic environments like our lakes, and they're free-floating instead of rooted like plants. But at the cellular level, blue-green algae are actually bacteria. So you can think of cyanobacteria as the more scientific, probably more precise term for them, but blue-green algae is a lot easier to remember because it floats around along with green algae that we recognize, and it lives in lakes and that sort of thing. So we'll continue to refer to it as blue-green algae, but it's important to remember that it is actually bacteria at the cellular level. So they're very, very small. Like I said, they're bacteria that photosynthesize. These organisms are found all over the world, from the oceans creating oxygen through photosynthesis, just like plants, down to our own agricultural fields right here in Indiana. Anywhere you see soybeans or other kinds of legumes growing that have the ability to fix nitrogen, bring it in from the atmosphere and down into the soil, it's actually blue-green algae in the roots that are doing that work. So these are very relevant, very important organisms. And they're microscopic, but can be found and seen in visible colonies and blooms. So when a whole bunch of them get together, that's when we'll start to actually be able to see them with the unaided eye. Blue-green algae are natural. Like I said, they're super important both in terrestrial ecosystems and in aquatic ones, but they can get way out of balance too. Some of these species produce toxins, such as microcystin, which is the main toxin that we'll be talking about today. And there are still a ton of mysteries to solve, particularly about aquatic blue-green algae, freshwater blue-green algae like we'd find in our lakes here. The pictures that you see on the side right now are all the same species of a blue-green algae. It's called Microcystis aeruginosa. And that first picture you see in the upper left is a microscopic picture. So these are individual microcystis cells that you see. And so when you're looking in the water, if there were just one or two of these cells, there's no way you'd ever see them. But when they start to clump up, like that picture just below, we start to see kind of globs of these colonies. And when the water is in perfect conditions for a blue-green algae bloom, when they can grow really quickly, we start to see them kind of taking over areas of the lake surface. Here you see in this picture on the bottom right, kind of that oil slick look. It's very shiny. It just looks like a part of the water, but you know it's not just a part of the water. There's something in the water that's kind of moving along with it. That is a blue-green algae bloom, or specifically a Microcystis aeruginosa bloom, but many of them kind of look like that. Those conditions that I was talking about earlier that they really, really love to grow in is warm water, very still water, and water with a lot of nutrients in it, especially phosphorus. They do really well along with other aquatic plants and algae when you have that kind of combination going on. But because they're very small, they can react really quickly to these different changes in conditions and can kind of take over a lake. When these huge populations of blue-green algae die off, so when they've used up all their resources and start to die off again, they sink to the bottom of the lake where they can decay. And this process also uses up oxygen and can be really bad for fish. So that's a couple of the issues that we're looking at with blue-green algae. Some more of those mysteries that we don't know are toxins are not well correlated with cells. So more blue-green algae doesn't always mean more toxin and vice versa. We'll get into that a little bit more with some of our research later. But right now we'll switch over to talking a little bit more about these toxins that they can produce. So this table right here is from the Idem algae toxin thresholds page, their blue-green algae page for their research that they do. Idem samples state parks and beaches for toxins and for blue-green algae, and we'll be kind of breaking down their program and looking at what their monitoring looks like here in a second. But these were standards put together from the World Health Organization, the US Environmental Protection Agency, and the Ohio Department of Health guidelines. So this is multiple agencies coming together to kind of craft guidelines based on human and also animal health, like you see over there to the left on the table. There are multiple blue-green algae toxins, like I said, but this one, microcystin, is going to be the main one that we're paying attention to because it is the most common in our state, and it is also the most common in our county, the most likely one that we'll see at all. It can be hard to relate to these numbers on their own, which I totally understand. Is 8 a lot, or a little, what about 0.8? So kind of having the context for those numbers. Later on in the presentation, we'll come back to some more of these numbers. But for more context, the unit of measurement for these things is in micrograms per liter. Another way to word this is part per billion. So you can imagine for every drop of water, or for every drop of a toxin like microcystin, there would be a billion drops of water. And that's kind of how potent or how relevant these toxins are. Thankfully, we don't get high numbers on these lakes very often. And like I said, we'll talk about that, but it's something that we're continuing to keep an eye on. We'll kind of zoom in on that table here just a little bit. The Human Recreation Advisory for state parks and beaches is 8 parts per billion microcystin. Recreation is prohibited at state parks and beaches at 20, and dog recreation is prohibited at 0.8. Dogs are much more susceptible to toxins like these, not only because they're smaller, but it hits some of their organs a little bit differently, and they're much more likely to consume it directly. Dogs don't really care what's in the water that they're drinking. They just are thirsty when they're at the beach. So they'll lick up any of that water, or they'll lick it off their fur. So they're more likely to ingest more of it in that way. Here, we will be talking about Idem's beach monitoring program and kind of what they do. And then in the second half of the presentation, we'll be talking about our Lilly Center protocols and our monitoring program that we've got going on right now to kind of compare and contrast the two. So Idem samples 18 beaches at 15 different state parks and state recreational beaches, plus the only DNR-owned dog park in the state. So dogs and this toxin, that's kind of a hot topic. They sample between Memorial Day and Labor Day, and all of these beaches get sampled at least once per month, and then they sample them a couple of weeks after a high count. So they'll kind of trigger another sampling if they get a high number and need to pay attention to it. Idem collects counts for blue-green algae species and for the total number of blue-green algae cells in the water. And they also identify those cells, which it's a lot of hours under a microscope, but they do so very quickly to get that data right away. They also sample for several algal toxins, microcystins, and the others that were on that same table. And they're specifically looking for high toxin concentrations or high cell counts. So there are some side effects or some health effects if you recreate in water that has a lot of blue-green algae cells in it. It can be irritating to the skin, and it can cause digestive issues and some more chronic health effects if you undergo prolonged exposure. So that's some of the things that Idem is paying particular attention to. They have 4 levels of risk categories when in Indiana. There's low risk, so that's where there's very, very little toxin or no measurable toxin at all. There's a beach advisory and beach caution, and then beach closure. So that's each step in the process that Idem would take in their sampling. In most years, Kasha let me know that in most years, we do not actually see a beach get entirely closed due to really high cell levels or really high toxin levels, which is great. This has happened only very rarely in the program's history. But in a typical year, Idem will see 2 to 4 lakes hit the caution level for variable lengths of time until conditions improve. So those are very dependent on temperature and nutrient inputs, how much rainfall is happening in a single year. So there's a lot of variables that play into these blue-green algae and toxin levels. But remember, blue-green algae doesn't always mean, a lot of blue-green algae doesn't always mean a lot of toxin, and very little blue-green algae doesn't always mean only a little bit of toxin. So that's why we have to sample both the toxin and the blue-green algae directly. So that's kind of a summary of Idem's program and the things that they're looking for. Do you have any questions so far? Caitlin, have we got any questions in the chat room? We'll give it a few. I'll give it a couple of seconds just in case people have anything that they're typing in. But if they do, you want to let me know. Cool. Take this opportunity to get a drink of water. Alrighty, let's get back into it. So here at the Lilly Center, we sample throughout the summer 12 all-sport lakes, the 12 all-sport lakes in the county, as well as Center and Pike Lakes. You can see the map there has all of those major lakes that we sample labeled, but our county has over 100 lakes, so there's quite a lot of those going on, and we're sampling the biggest and the most publicly recreated on typically. We also sample 7 public beaches, swimming beaches, across 6 different lakes. And we do this sampling weekly from June to mid-August. So we're focusing on that high recreation period, as well as that same period being when blue-green algae is most commonly growing. We sample for a number of things, including algae populations. So we collect a water sample that we will later analyze to identify the populations that are present and how many cells are in the water at a given time. We also sample for microcystin toxin, specifically microcystin because we know it's the most common in the area. I'll talk about that a little bit more too on a later slide. And water conditions. So an important part for understanding blue-green algae and toxin production is knowing what the water is like. What is their environment like while they are living in our lakes? So that includes everything from dissolved oxygen and water temperature and pH to nutrient concentrations throughout the water column. So those are all things that we sample on a weekly basis. That's that regular schedule that I mentioned down there. We sample them weekly on the same day, same time of the week. But we're also working on getting some algae bloom specific sampling going on. So that would be opportunistic. When we hear about a bloom or when we see a bloom, we would jump on that to get it sampled and to see what's going on. It's important for us to see the lakes in both conditions so that we can understand when algae are changing what they're doing. Are they producing toxin at certain times and when certain conditions hit, or is there another factor that we aren't yet seeing? So that's a continued toxin monitoring program that we're having going on weekly throughout the summer. Previous research definitely helps guide all of our efforts, as good research does. So there are a couple things that we know ahead of time. Blue-green algae and microcystin toxin are relevant in Kosciuszko County. Even anecdotally, we are getting emails and phone calls from people every once in a while, especially as we start getting into July and early August, as temperatures continue to go up. We start to hear about some of these blooms. In fact, the picture there is on one of the lakes in our area from last summer. So just kind of localized bloom, that greenish texture that you see on the top of the lake scattered amongst the lily pads there. So we know that these things are relevant in Kosciuszko County. We did a pilot study back in 2009 during which lab analysis of a sample from Lake Wawasee measured 63 parts per billion of microcystin. If you remember that Idem guidelines table from a number of slides ago, that's 3 times higher than the human recreation prohibited level for state waters, which is 20 parts per billion. So we know that doesn't happen very often as we've been continually sampling, but we know that it can. And so of course we want to be paying attention to these numbers and to sort out ways we can know when water is safe and make good decisions about recreation. We also know, like I said before, that algae populations and toxic conditions vary between our lakes. And I'll get into that too. I've got some data for you to see that I'll kind of walk us through, but each of our lakes here in our county are different in terms of the conditions that they're experiencing within the water and the populations that are present. So we can't treat all of our lakes exactly the same, and we can't just sample one lake and project it out to the rest. They all act a little differently. We also know, due to some research from previous years that we've done, that forecasting and prediction are not quite yet feasible methods for determining water safety. We don't yet have something that we can quick dip in the water and take a look and see if it's safe. This is because we're still not sure, and researchers all over the world are still not exactly sure why these toxins are being produced and what triggers their production and their release into the water. So we were trying to answer the question, can we determine if the water is toxic by measuring something other than the toxin? And so far the answer is not yet, but we're still hopeful that that's possible. So let's get into some of that data to look at our lakes specifically here in the county. This is a big table. Let me break it down, walk us through it. Don't worry about paying attention to every number on here. I'll highlight some things a little later on to kind of draw our attention to some of the main takeaways for this. So these are the averages of a number of parameters from June to mid-August of the past couple of years, the past 3 years, 2015, 2017, through 2017. SD is Secchi disk. That's a measure of water clarity, seeing how far down into the lake we can see, and that's measured in meters. MC stands for microcystin, and that's in parts per billion, like we talked about earlier. So like we said, 8.0 being a human recreation advisory level for Idem, we're seeing these numbers are much lower than that for humans. So that's good news, like I said earlier. Then we also see blue-green algae. That's all species of blue-green algae in the number of cells in the water per milliliter of water. And then the last column over there on the right is the percentage of blue-green algae of all of the algae in the water. So that includes green algae, diatoms, those sorts of photosynthetic organisms living in the water. That's how dominant blue-green algae is in our lakes. So let's pay attention to a couple specific things on this column. I will use my pointer to take a look at these. So we can see very clearly that the relationship between toxin and microcystin, or between toxin and blue-green algae populations is a little interesting. Big Bar B, for example, has relatively low microcystin for these years, but it has relatively high blue-green algae counts. The amount of blue-green algae in the water is relatively high. The same goes for Winona, where we see low, again, some of the lowest microcystin across these particular lakes, but the highest blue-green algae counts throughout the year, and very dominant of all the populations. The inverse is true for Dewar and Wawasee over these years. They had slightly higher microcystin on average, but much lower blue-green algae for both of these lakes. So there also isn't a clear pattern north to south, east or west across the county either. So this is something that's very lake specific, and it has probably a lot to do with what the community of algae itself is looking like, which species compete well at what times, and the different pros and cons and strengths and weaknesses of different species. So we are working on zooming in further to pay attention to what key species may be having an impact in these lakes in particular, and what the water, the algae, and the toxin levels are all looking like when the water is undergoing a bloom and not just during these regular weekly samplings. So this is us zooming in to learn more about blue-green algae and microcystin. So what about these bloom events? Bloom events are key for public health and for research both. People want to know when their water is safe so they can make good decisions about recreating. And we also want to know when the water is blooming, when blue-green algae are up and at them, and when they're really proliferating in the water. So everybody wants to know what's going on, including us. So the next portion of this talk is going to be about identifying blue-green algae blooms. So identifying the things you don't have to think about, the things that are not of concern, and then blue-green algae as it relates to your health and determining when to jump in the water and what to do with that. So we will get right into it. We're going to start with what isn't blue-green algae, which is a lot easier than just starting fresh with what is. So if the green stuff that you see on the surface of your lake has roots, leaves, or discernible tiny individual plants, it's not blue-green algae. Duckweed and watermeal, like I have pictured here, duckweed is that top left and watermeal is the bottom right. These are types of tiny, tiny aquatic plants that can cover the surfaces or shorelines of ponds and lakes. They can just absolutely form this huge mat across the lake so that it looks like there's something growing in the water, on the surface of the water itself. And in fact, that's what they're doing. But these, you can tell because of their size, even though they're tiny for plants, they would be too big to be blue-green algae. So neither of these are blue-green algae. These get a little tougher. If the thing that you're seeing on your lake is stringy or hairy, if it's got filaments, it's filamentous green algae, most likely. This organism is bigger, not quite microscopic. You could see and pull apart individual pieces of these algae, and they'll usually drape over a stick, like you see in that picture right there. They'll kind of glob together like that and just be kind of knotted together. So that's green algae. There's a number of different types of green algae, but in general, this is a really good trick to know that if it's sticking together like that, it can drape over a stick. It's likely green algae instead, which no toxins, it's just like a plant, just kind of growing and doing its thing. The one on the right is a little bit trickier. That you can see that the particles are super tiny and they seem to distribute in the water almost like a liquid or almost be a part of the water or across the shoreline. But we notice that it's yellow, so the color is one thing, and that it's kind of dusty in texture. This in particular is tree pollen. This is what tree pollen looks like when it gets into our lakes. Because it's a lot smaller, it'll act a little bit more like blue-green algae in the water because it's tiny, but you'll note that color. And I believe it will also sink if you put water on top of it, it'll sink down. It won't float quite as well as blue-green algae. So that is tree pollen, not blue-green algae. But here we have an example of blue-green algae. Because blue-green algae are bacteria, remember, for cyanobacteria, and not a true plant or algae, they hang around in the water a lot differently than a plant or an algae would. They're tiny, tiny cells, right, that are only visible to the naked eye when they're densely gathered together, like in this picture here. They seem to almost form a part of the water. You can see here there's streaks where the wind kind of blows through it gently. There are patches that seem a little darker or more condensed. But if you were to take a stick, you'd probably just swirl this around and wouldn't be able to pick up what you would recognize to be individual cells or colonies. So this is what a typical blue-green algae bloom would look like, especially for the kinds of species that we're looking at here in Kosciuszko County. Here's another example. Sometimes there are so many blue-green algae cells and they are just being continually moved throughout the water column that it makes the entire body of water, or at least at that shoreline, seem like a different texture entirely. We'd call this kind of a pea soup appearance. And you can really notice this when the water is in motion, kind of like this picture is showing here, that it's just totally distributed throughout the water and is moving in all of the same ways that water does because they're absolutely tiny little cells. I actually have a video here to show that you can see some of this in motion. This is from California's equivalent of Idem, their Department of Environmental Management. So I'm just going to navigate over to that real quick. And I'll have it on mute and only show a portion of this video because the main thing is that you see kind of how it moves along with the water and how it colors the water. Yeah, you can see it kind of just changes the tint of the water slightly. It might change the texture if it's really, really thick. You can see kind of across that whole right side over there that it seems really dense, but it moves very easily with the water. This is something that's fully integrated into the lake that you're looking at. There's a really cool image here in a second, under the water. You'll see these kind of globs floating around in the water column. These individual cells can glob up and kind of grab onto each other just a little bit, kind of stick together a little bit. And that's where you might see what you think might be green algae because it is kind of clumped up in that way. But it's going to have that very globby texture, and it's going to be kind of hard to pick it up because it's just going to be right there with the water and kind of escape your hands along with the water too. So that's some examples of a blue-green algae bloom in motion. Come back over to our PowerPoint here. Oh. Alrighty. There we go. So now, if you see a blue-green algae bloom, like I said, that helps you. If you can positively identify a blue-green algae bloom, helps you make a smart decision about recreating. You can decide whether or not you want to jump in, or allow your animals to play in there, that sort of thing. But we'd also love to know when that is occurring so that we can learn more about when these blooms are happening, what the water is experiencing at the time, and that sort of thing. So we actually have a handy dandy window cling that our wonderful public relations specialist, Abby Finney, designed and put together for us, that you can just, I believe we'll have a follow-up survey from this webinar, and in that you can include your address and we'll mail one of these to you. It's just a window cling that will stick right on the window of your house or of your boat maybe while you're out, and it kind of walks through what information is most useful for us, what information we need to accurately and quickly respond to an algae bloom when it happens. This also gives you something that you can actually do with that knowledge aside from just knowing whether or not you want to swim. This helps that information go somewhere, and we're really appreciative for that. So first of all, we'll kind of walk through these individually. The date and time of the bloom, the date and time that you're observing the bloom. These blooms can dissipate really quickly. Sometimes these water conditions are changing and populations are changing. So it would be really helpful to notify us right away when you see these things, including the time that you're looking at them. That way we can also see the difference between the time when you saw them and when we get out there. Next is the lake name and location or home address where the bloom is being observed. So if you're looking off of your own pier, or maybe you're at a friend's house, they have a lake property, or maybe you're out boating, you're out in the middle of the lake. We need to know which lake it is specifically, and location or someplace that we can navigate to access the water specifically. That is very helpful. Third, a description and hopefully a close-up picture of the bloom. This helps us very much to be able to determine if what we're looking at is a blue-green algae bloom, or maybe at what stage of blooming that it is. All these things are very useful, and we actually do hold on to those pictures and include them with our data, so we have some visual data to go along with our numbers and measurements that we take. A description can be anything from, oh, it has a pea soup appearance, to it's making tiny globs in the water and that sort of thing. Other pictures or descriptions of the extent of the bloom, so how far the bloom is going along the shoreline or across your lake, is also very helpful, so we can tell how far reaching these impacts might be. Lastly, any general comments or observations. Smell can be actually a really helpful part of that kind of observation. Does it smell fishy or sudsy? Are there like, does it kind of smell like decay at all? And if decay, plant decay, or kind of other biological decay, however your nose decides to describe that is actually really helpful for us. Any other observations, say there's dead fish further along the shoreline, that sort of thing. All of that is very relevant information to us as we decide whether or not we can go out and sample. Last step is gathering all of those observations, typing them up, and emailing them to the Lilly Center, lakes@grace.edu. Not too complicated of an email address, but that'll get forwarded on to us or my team. And then we'll be able to make a decision about going out to sample it and be able to learn more about our lakes under these conditions. Ultimately, if you aren't sure if it's a blue-green algae bloom, if you're like, I went through that whole IDing thing, I'm just not sure, take pictures and shoot us an email anyway. Like we've described, there's a lot we still don't know about blue-green algae, when they produce toxins and when they don't. So we would appreciate any information or observations that you have on your lake. And to help us to get to the bottom of those mysteries, we want to monitor the water not only each week, but in the summer, but also when it's undergoing blooms. So next, I'll post one of the pictures of a blue-green algae bloom that we've seen, and we'll kind of walk through how I might fill out this sheet if I had walked up to a pier and saw this at the end of the pier. That's kind of an example, like a practice report of sorts. So let's pretend that or a whole group of us maybe, let's pretend it's all of us, that'd be fun. So a whole group of us are on the public boat launch at Winona Lake, say. This picture was not taken at Winona Lake, but we're gonna pretend that it is. So this is our lake, we're about to go out and have a great day, and we observe this in the water. We see that the water has that kind of shiny look to it, just like water normally does when it's still, but just under the surface or just at the surface, there's that green swirly look. Can't easily discern any leaves or roots. And it seems like when we swirl it around with a stick or something, it wouldn't be able to be picked up by that stick. It's just gonna move right along with the water. Because of all those things, and also it's not yellow, it's definitely green or blue-green. I'm kind of thinking that this is a blue-green algae bloom. So we look at the date and time. Caitlin, what's the date and time?
20th.
Okay.
1:37.
July 20th at 1:37. So I'm going to pull out my phone and open up my email app and type that in. The lake name, where you said we're at Winona Lake and we're near the public boat launch, which would be enough to be able to get my team there, or the Lilly Center's team over there to take a look at it. That's a specific enough location. Description, so I'll say it kind of has that blue-green color and it swirls around in the water, and it looks pretty big, like a pretty vast extent of a bloom. It's not quite a pea soup appearance, but I do see it kind of as a scum up there where the boat launch meets the water. It seems like it kind of piles up there. I take this picture with my phone, attach that with it, and then any other comments or observations. We do see a couple of fish down the shoreline that are dead. That's not a great sign. We'll include that in there. And we'll say, doesn't particularly smell like anything though, so we'll include that in there too. And that is a blue-green algae bloom report. We're, type in the address, lakes@grace.edu, and send it off. And that is how that whole reporting process would go. Like I said, these clings, totally free. Just include your address, your mailing address, in the post-webinar survey that we'll send out to the email address that you signed up for this webinar with, and we can get one of these to you easy peasy. That's not the only thing you can do though, for sure, about blue-green algae. There are more steps that you can take. First and foremost, one of the most foundational things is reducing the amount of nutrients that enter into our lakes. Like I said, these blue-green algae really love warm, still water, especially when they have all the food that they need to get going. They photosynthesize, but they also take up other nutrients as well, and are competing against other good algae and other good organisms for those nutrients. So if we bring those nutrient levels down, we can help keep our lakes clean, healthy, safe, and beautiful. Nutrients get into our lakes by fertilizer on lawns or grass clippings, or excess leaves getting into the lake, by the feces of animals, including geese or dogs, others, and that sort of thing. Storm drains, when they're all blocked up on your street with leaves or grass clippings, all of that will go directly into a body of water. Those things will decay, they'll use up oxygen, and then they will release their nutrients for blue-green algae to take advantage of. So reducing those nutrients really helps protect our lakes quite a bit. To emphasize, to multiply those efforts, getting connected also helps a ton. If you have a lake association, or even just getting to know your neighbors on either side of your property, joining together really amplifies these efforts across our whole lake shorelines. So a healthier lake for everybody means safer recreation, a more beautiful lake, healthier fish, and honestly, better property values. A cleaner, more clear, beautiful lake really helps everybody want to be there and enjoy the lake too. Other quick tips for you. If you're not sure if blue-green algae kind of freaks you out a little bit, even if the water looks clear, one thing that Idem suggests, and that we would also suggest by Idem's recommendation, is rinsing off after recreating. The main recommendation is warm soapy water to do the most good. You can also rinse off your pets, so it's not staying on their fur, that sort of thing. That'll really get any other, blue-green algae or not, other irritants, that sort of thing off of your skin. which is great. And if you're in doubt, stay out of the water and you can pick another day. That's a little clearer, a little more straightforward. So with that, do we have any other questions in the chat bar? Awesome, sounds great. Oh yeah. Mm-hmm.
Okay, yes, we do have a few questions. Let me get up to the top here. Debbie asked, do you send the samples off to a lab for testing, and can you talk about the testing process?
Yeah, that was an awesome question. Thank you so much for asking that. So microcystin is one thing that we sample here in-house. We did it last summer and this summer as well. We're doing so under the same kind of QA or the quality assurance protocol that Idem uses to make sure that we're collecting good data. So when we gather a sample from a lake, store it in a freezer, and then we freeze and thaw it a number of times to break open the cells and release all of the toxin that might be in the water. And then we run that through a system of chemical reagents. The long story short of that process is that the water will turn a certain shade of color based on how much toxin is in there. So we run all of our samples for the week that we've gathered on Thursday. And then we have all of that data on Friday to be able to see what our lakes are looking like. Collecting blue-green algae samples is a little different because those have to be identified and counted by hand under a microscope. So that process, we gather some water, and when we sample for both algae and microcystin, we are sampling the top, essentially the top 6 feet of the water, because that is what people will more likely be exposed to. So we're taking a sample of that whole 6-foot range in the top of the water column. And so for algae, we're collecting that, we're preserving the samples, and then mounting them on a microscope slide, making a permanent slide. And then we can go in and identify what species are present, and how many cells are in the water. When Idem does this process, they've got a little bit of a different process because they are looking for a really quick assessment, really fast assessment of how many cells are in the water. This is a long process. These things are very small and they look very similar. So, so they do kind of a rougher, not rougher because it's still a very valid way to do it, but they're going very quickly to identify how many tens or thousands of cells are in the water at once, so that they can report that right away. Our algae work is a program that we are still in the process of building up and really making robust and strong, so that's not something that we do on a weekly basis yet. But we're working towards speeding up that process as well. And then those numbers are included in some of our bigger reports, bluegreen algae reports. Yeah, I hope that answered your question on that. And if it didn't, feel free to ask again, kind of re-clarify.
Okay, we had a few people ask who can report. Is it specific to Kosciuszko County?
Yes, so that's a good question too. Our focus here at the Lake Center is specifically Kosciuszko County. So the lakes that we would be able to go out and respond to reports about, and the data that we would be including in our reports is going to be Kosciuszko County only. That was one of the things, one of the questions that I was hoping Kasha would be able to answer for us is whether or not other waters would should be reported to Idem. They have a form that covers blue-green algae blooms, that you can fill out and send in, especially if there are potential health effects. So that's something that I can reach out to Kasha about to clarify for examples outside of our county. For other counties might have other organizations doing this. There's not very many in Indiana, but other states might as well have county or state level organizations that are doing this kind of work.
If we find that link, maybe we can send it afterwards.
Yep.
Mm-hmm.
Yeah. So then afterwards.
Kendall asked, are there algae blooms that are similar but a slightly different color that are not cyanobacteria?
Ooh, yes, there are. There are some kinds of algae or other free-floating microscopic organisms that can be other colors, like red. I think on Center Lake a couple of years ago there was a red bloom at a weird time of year that we actually did go out and sample and learn about. So I guess that's a good point of clarification too. If you see something very weird that does not quite fit into what we've described as a blue-green algae bloom, but especially if it does seem like an algae bloom that's like really thick in the water and that sort of thing, when in doubt, go ahead and shoot us an email anyway, and we'll definitely take a look. But yes, it is possible for other kinds of blooms to be other colors. Predominantly, you'll see green. You might see green with a white scum on top. As the blooms decay or get mixed around, sometimes they can kind of foam up, and that can still be, even though it might have a slightly different color, that can still be blue-green algae as well.
Okay, we have, for those who signed in a little bit late, can you explain the difference between cyanobacteria and blue-green algae once again?
Yes, for sure. So the difference between cyanobacteria and blue-green algae is nothing. There is no difference between those two. Those are two names for the same organism. Cyanobacteria is probably the more scientifically understandable name. It is cyano, like cyan, the color, kind of that blue, that kind of vibrant blue color, and bacteria, which these organisms are actually bacteria, but they're bacteria that can photosynthesize. We call them blue-green algae kind of anecdotally, or it's a more of like a common name versus a scientific name, because they're free-floating. It's a little easier to understand them as algae because they compete with green algae, which is more the technical definition of algae. But yeah, cyanobacteria and blue-green algae are the same. And they are natural in our waters here, but they can get out of balance and outcompete other forms of algae.
Just to kind of follow up on one of those points you made earlier, Kendall says at one of the lakes festivals, there were several samples of greenish algae, and the message was to show how to tell if it's a dangerous green or not. Should we assume if it's green that it's cyanobacteria?
The color is probably less likely to be the indicator. I mentioned earlier, there's tree pollen that can look yellow, and green algae will look green, and blue-green algae, here's something that I didn't mention on that previous slide. The blue part in particular is for a specific pigment, phycocyanin, but blue-green algae also have chlorophyll, so they can have both. The blue part is more of a chemical definition than maybe a visual one. So if you see a bloom and it looks just straight up green, but it has all of the other characteristics of a blue-green algae bloom, and it's probably blue-green algae. The blue part is more indicating that chemical compound that we would identify more with blue than with green. So they can just look green. They're not going to necessarily look teal or something like that. Mm-hmm.
Okay, Debbie asks, what would you suggest other lakes do to have a testing protocol?
That's a great question. I would see if there are any groups in your area that do algae identification or microbiological identification. Sometimes public health will do this. Water treatment plants will sometimes also do this. So different organizations like that. To see, I would start by seeing the prevalence in your area. Now, if you're already seeing blooms, if you're already in the summertime getting occasional blooms, then you know it's probably something that, that your lake will be working on, or a group could start working on. There are a number of quick testing methods to determine if it's blue-green algae or not. Based on, there's one called the jar method, where if it floats, it's blue-green algae, and if it sinks, it's green algae, and something like that. That is something the literature research we have not gotten into yet, but it's on my desk right now to take a look at, because we'd love for people to be able to make a quicker assessment about what kind of algae it is. That stick method, like I mentioned before, you can dip a stick into the water and lift it up. That will show you very clearly, at least if it's green algae, because if it's going to drape over the stick quite a bit and almost look like hair, that's green algae. Different forms of algae, blue-green, that's cyanobacteria, and green algae can be present all at once. They can be present right alongside other aquatic plants like lily pads. So you just want to be able to tell if you're having something where there's a lot of things going on at once, or if you just have a big bloom, that sort of thing. Yeah, so I would start there. I would start with figuring out if it's prevalent on your lakes, and then start looking at quick methods for determining those things. Awesome. Well, if there are no other questions, I've got a couple links here shown on the PowerPoint. These links will also be provided to you in that post-webinar survey. So don't feel like you have to jot them down right now unless you want to. Hey, if you're super curious and want to get into this stuff right away, right now after the webinar, that's awesome. This is a link to our own Bluegreen Algae research page where you can learn a lot more and maybe take your time with it if you are more of a reader than an audio learner. That's a great place for that. And this is the link to the Indiana Department of Environmental Management's algae page, specifically where they have more of their numbers and such listed as well. Lastly, please do fill out that survey, and if you're interested in one of these clings to help us see where blue-green algae blooms are happening here in Kosciuszko County, just include your address on that form, and we're good to go. Thank you so much for joining us and for your awesome questions. I really hope that you can enjoy the lakes this summer, and yeah, thank you very much.